Welcome back. Today, we are jumping straight into the heart of biology's most famous and honestly awe-inspiring partnership. We're talking about life's ultimate data storage system and its unbelievably versatile builder. Yep, I'm talking about DNA and RNA. In this explainer, we're going to completely demystify these two molecules. By the time we wrap up, you'll know exactly how they work together, and more importantly, how to spot the key differences between them. Here is a quick look at our roadmap for today. We'll kick things off with the molecules of life, then move into spotting the differences. After that, we'll look at translating the code, and finally, we'll wrap up with gene editing and epigenetics. All right, let's get right into section 1, the molecules of life. To start, I want you to picture something. Imagine you have the most complex, precious source code on the planet, literally the instruction manual for building a human being from scratch. If you just leave it out in the open, it's going to get corrupted, right? Now, we've all seen a digital blockade like this one while browsing the web. You know the drill. This website is using a security service to protect itself. You try to access a page, and a firewall stops you cold, thinking you might be a malicious bot. It's doing its job, protecting the source code. But what's absolutely crazy is that nature invented this exact same concept billions of years before the internet even existed. It's a perfect metaphor, really. Just like websites rely on digital security protocols to block malicious code from tanking their servers, bacteria have their own molecular security system. They use these things called restriction enzymes. You could think of them as a microscopic immune system that actively identifies and chops up foreign, malicious viral DNA to protect the bacterium's own genetic code. Whether we're talking about a massive server farm or a tiny single cell, the prime directive is exactly the same: protect the data at all costs. So what exactly is this data? Well, it's DNA, or deoxyribonucleic acid. It's a long polymer coiled up into that beautiful, iconic double helix, and it carries the genetic instructions for the development, functioning, and reproduction of basically every known organism. It is the master blueprint, and because it's so unbelievably precious, in complex organisms like us, it's locked away deep inside a vault called the cell nucleus. It's heavily guarded and incredibly stable. But here's the thing. A blueprint locked in a vault can't build a house all by itself. It needs a partner on the job site. Which brings us to section 2, spotting the differences. You can think of this as the alphabet of life. Just like your computer uses combinations of ones and zeros, DNA and RNA use simple chemical units called nucleotides. We've got adenine, cytosine, guanine, and thymine, A, C, G, and T. But here is the absolute most crucial distinction to look out for. DNA relies on thymine, but RNA swaps that out for uracil. So if you're ever looking at a genetic sequence and you spot a U, that is your immediate dead giveaway that you're looking at RNA, not DNA. When we put them side by side, the differences really pop. DNA is double-stranded, that famous twisted ladder. It uses a sugar called 2-deoxyribose, and it is strictly built for highly stable long-term information storage. It's the archival hard drive. RNA, on the flip side, is single-stranded. It uses a standard ribose sugar, and like we just mentioned, it substitutes uracil into its alphabet. Because it's only a single strand, it's incredibly versatile. It can fold into all sorts of complex shapes and actually move around the cell. So while DNA is the static archive, RNA is the active messenger and the tiny machine that actually translates the code into physical reality. Let's look at exactly how it does that in section 3, translating the code. 3 billion. Let that number sink in for just a second. The human genome contains approximately 3 billion base pairs, packed incredibly tightly into the 46 chromosomes in almost every single one of your cells. To put that into perspective, if you unravel the DNA in just one of your cells, it would stretch about 2 meters long. That is a massive amount of data that has to be carefully and flawlessly decoded every single moment you're alive. So how on earth does the cell pull off this logistical miracle? Well, it all comes down to an elegant 2-step dance. Step 1 is transcription. Let's say the cell needs a specific instruction. Enzymes will actually unzip a tiny portion of the DNA double helix, and an RNA copy is made of just that one specific gene. We call this copy messenger RNA. Then comes step 2, translation. This newly minted, single-stranded messenger RNA leaves the protective vault of the nucleus and travels out to a cellular factory called a ribosome. The ribosome acts like a scanner, reading the RNA sequence 3 letters at a time and translates it into a chain of amino acids. And those amino acids, they fold up into proteins. This whole process is happening inside you right now, literally as we speak. The genes, made of DNA, hold the raw instructions. RNA acts as the messenger and the active translator, but the proteins are the tiny physical machines that actually get the work done. They're digesting your breakfast, they're firing the neurons in your brain so you can understand what I'm saying right now. They're repairing that paper cut on your finger. This flow of information from DNA to RNA to protein is the absolute fundamental mechanism keeping you alive. Which leads us perfectly into our final segment, Section 4, Gene Editing and Epigenetics. There's this fantastic quote from a Guardian article on epigenetics that I absolutely love. It describes the concept so well. Quote, "This is like a pink highlighter telling the cell, hey, this part's important." You see, to fit inside the cell, your DNA is tightly wrapped around proteins called histones, kind of like threads spooled around a bobbin. Epigenetics involves tiny chemical tags that attach to these histones. These tags can actually loosen the DNA, making it way easier to read, acting exactly like a pink highlighter on a textbook page. Or they can tighten the spool up, hiding the code and effectively turning the gene off. But here's the really wild part. This pink highlighter isn't actually changing your core DNA sequence at all. It's just changing how the cell reads the blueprint. And even more fascinating, these epigenetic marks don't just cluster on the DNA. They also modify RNA as it travels around the cell. This creates a deeply complex secondary layer of biological control. It dictates not just what genes you have, but precisely when, where, and how intensely those genes are expressed. Now, if epigenetics is highlighting the text, CRISPR is basically physically cutting and pasting it. Take a look at this. Notice how the very first component of the CRISPR system is single-guide RNA? Scientists are essentially hijacking RNA's natural ability to hunt down specific genetic sequences. We can program an RNA guide to find an incredibly precise spot within that massive 3 billion letter DNA genome. Once the RNA guide finds the exact match, it anchors the Cas9 enzyme in place. Cas9 then acts like a pair of molecular scissors, making a precise cut in the DNA strand. From there, we can literally knock out a faulty gene, correct a mutation, or even insert an entirely new trait. It's honestly staggering to think about. We have gone from simply inheriting this master blueprint to actively programming the builder. And I think that leaves us with a really profound, provocative question to wrap up our explainer today. Now that we understand the critical differences between how DNA strictly stores the code and how RNA actively translates and guides it, how might our newly found power to edit this dynamic duo alter the very future of human evolution? Think about that for a bit. Thanks so much for joining me. Keep questioning, keep learning, and I'll see you in the next one.